Hello everyone, welcome to Science Made Easy. As I said in the last video, in part 2 we will be answering some more questions. Here is the part 2 about development of female gametophyte. In part 1 we have seen development of male gametophyte. The link is provided in the description. Make sure to watch the video. Coming to the part 2, what is the female gametophyte? The embryo sac is usually called the female gametophyte. Let us see some parts belonging to gynoecium. The carpels make gynoecium. These carpels are also called as megasporophyle. Each carpel consists of three parts: ovary, style, and stigma. In this, the ovary has ovule, which is also called as megasporangium. These are the parts of gynoecium. This is stigma, this is style, this is ovule, which is inside the ovary, and this is the ovary wall. These parts make the gynoecium. Let us see some more details about ovule. Ovule generally originates from the inner surface of base of ovary. It is attached to the placenta with a stalk which is called as funicle. The point where funicle is attached to the ovule is called as hilum. The funicle grows beyond hilum through the body of ovule and forms a ridge which is called as raphe. The ovule has parenchymatous mass of tissue which is called as nucellus. The nucellus is covered by integuments of one or two coverings. These integuments do not completely cover the nucellus and leave some portion which makes the micropyle. These are the parts of ovary and ovule. This is the hilum, the point of attachment of funicle. Funicle is the stalk through which the ovule is attached to the placenta of the ovary. Micropyle is the portion where the integuments do not cover the nucellus. This is the micropylar pole and outer integument and inner integument of nucellus. This is the nucellus, the tissue of the ovule. The embryo sac which is formed inside the ovule. The chalazal pole, this is opposite to the micropylar pole. These are the different parts of ovule. Ovule is the megasporangium which helps in the formation of megaspores. The development of female gametophyte occurs in two stages: megasporogenesis and megagametogenesis. Megasporogenesis. The formation of megaspores from megaspore mother cell in megasporangium is called as megasporogenesis. Megagametogenesis. It is the formation of female gametophyte from megaspores is called as megagametogenesis. In the further video, we shall see these processes in clearly. Megasporogenesis. Firstly, an archesporal cell undergoes differentiation and forms the megaspore mother cell from where the megaspores originate. Let us see the differentiation of archesporal cell. The primary archesporal cell, which is also called as hypodermal initial. There is a single hypodermal initial. It undergoes periclinical divisions. The primary archesporal cell is present at the apex of nucellus. The nucellus is the parenchymatous tissue inside the ovule, and at the apex of nucellus is the archesporal cell. This differentiation gives rise to different tissues. They are outer primary parietal cell and inner primary sporogenous cell. The outer primary parietal cell forms the primary wall of the cell. It may or may not undergo further divisions. The inner primary sporogenous cell directly behaves as megaspore mother cell. This megaspore mother cell undergoes meiosis and forms four megaspores. These four megaspores are haploid in nature. If you see the image, this is the nucellus, which is a parenchymatous tissue inside the ovule. At the apex of the nucellus is the archesporal cell. The differentiation of this cell produces different tissues that give rise to the megaspore mother cell, and at last the four megaspores originate from megaspore mother cell. Clearly, this is the megaspore mother cell which has originated by the differentiation of archesporal cell. It undergoes division 1, that is meiosis 1, and produces the megaspore dyad, and the meiosis 2 produces the megaspore tetrad. This is the process of megasporogenesis. Megagametogenesis. It is the second stage in the development of female gametophyte. In this process, the female gametophyte is formed from the megaspores. The megaspores that are formed as a result of megasporogenesis are arranged in axial row, forming a linear tetrad. If you see this image, the megaspores are arranged one below the other, forming a linear tetrad. Of these four megaspores, only one is functional. It grows at the expense of other three. This is the functional megaspore, and these three are degenerating megaspores. The functional megaspore enlarges in size and undergoes three mitotic divisions to form embryo sac. This embryo sac is 8 nucleate and 7 celled. The other three megaspores degenerate. Let us see the formation of embryo sac from functional megaspore in detail with the help of this image. Firstly, the functional megaspore enlarges in size and undergoes mitosis. After first mitosis, it enters the 2 nucleate stage. After second mitosis, it enters the 4 nucleate stage. After third mitosis, it enters the 8 nucleate stage. At the 8 nucleate stage, all the nuclei in the embryo sac are distributed throughout it, and the two polar nuclei reach the center of the embryo sac. They fuse and form the central cell. This is the reason why the central cell of the embryo sac is diploid, as it is formed by the fusion of two haploid nuclei, and the nuclei near the chalazal end develop into antipodals, and out of the three nuclei near the micropylar end, one develop into egg cell and the other two develop into synergids. So finally, the 8 nuclei in the embryo sac are formed in this way. The three near the chalazal end develop into antipodals. The three near the micropylar end, one develop into egg cell and the other two develop into synergids, and the central cell, which is formed as a result of fusion of two haploid nuclei. And this is how the female gametophyte is formed. I hope you found this video useful. Thank you for watching it. If you haven't subscribed to Science Made Easy, subscribe and please keep supporting Science Made Easy.